Сега малко тук може да стане, не съм сигурна. Сега тук се натовари. Hello. Hello, students. Това искам да бъде unmuted. Добре. Да, затвори. Hello, students. Do you hear me?
Hello, Professor. Yes, we hear you.
Yes, okay. I see several people here. Are we waiting maybe until 5 minutes more or what?
Yes, let's start at 5.
Yes. I don't know how to click on my camera, but I hope you hear me, okay?
We hear you very well. It's not necessary.
It's good. Okay, so we wait 5 minutes more.
Okay, then we start.
Let me see how to make it on a big screen. Did you see? Are you seeing a big screen now?
Yes, yes.
Is it this one?
We see the first slide, autoimmune and
Yes, this is
autoimmune.
the first slide. The thing is, it's not the full screen. Yes,
It is for us.
it is for you. Okay. Let me see. How can I do it here for me also? It's all right. Yes.
Yes.
Is it better?
It's the same for us.
Okay, it's the same for you. For me, it's a bigger one. Okay, please, when you hear during the lecture, don't hesitate to ask me questions if you want.
Okay.
So we could make a more better discussion.
Okay.
I'm very sorry to lose the first three lectures, but it was not my fault. The problem was with the organizer from the university with this model thing.
Mm-hmm.
So we should find a way. If you want, we could make more lectures. We have 2 weeks until the 7th week. Yes, 2 more lectures. And we will have them when you are finishing your exercises. I mean practicals. This week
We will finish the next week. Yes.
you will finish next week. Okay. Yes. So we have possibility to have today and the next week lecture, and the week after it may be no point to have it after you finish the course, but we could do it the next week also.
Mm-hmm. Okay.
Shall we start or wait a little bit? Please tell me.
We can start and they can join when they are ready.
So today we lose the first lecture. It was about overview on the immunity, and we start today from the autoimmunity and autoimmune diseases. I think this will be more helpful for you, and that's why we start with it today. So today we will talk about reactivity, which is the normal reaction, and autoimmunity, which is a pathological reaction. Then we will have more proper information about the autoimmune tolerance, then how it is appear to be dysregulated to become autoimmunity, which is the pathological state, and then some mechanisms of tissue damage which are characteristic for the autoimmune diseases, and how these autoimmune diseases appear. So we start with what we call autoreactivity. This is a term which is accepted to express the phenomena connected with the recognition of self-antigens which are into the organism all the time, and the induction of a reaction which is not damaging for the organism. So it's called autoreactivity, and this autoreactivity is a normal reaction in the healthy immune system. When in certain conditions, these reactions are changing, are dysregulated, and then we go to the state of autoimmunity, which is a pathological state, and it's a base for induction of different autoimmune diseases. About 10% of all autoimmune reactions which appear in certain conditions, and we will see later on, became chronic damaging for the organism and express clinical symptoms, then in this case we are speaking about autoimmune diseases. So you see there is three terms. Just a moment, please. We have three terms which are connected with reactions of the immune system against self-antigens, and these are autoreactivity, autoimmunity, and autoimmune diseases. They are not the same or do not cover each other all the time. So autoimmune diseases are chronic conditions with certain clinical picture and activation of different responses. So if we have a definition for normal immunity, which means normal immune reactions, there is normal antigens, there is a specific T lymphocytes or B lymphocyte cells which recognize this antigen, and then most often it might be bacterial or some other microbial antigens, which leads to activation and of the after recognition of this antigen. What is happening is activation of these cells, proliferation and differentiation. And this is the normal way of one immune response is going. But where there is a self-antigen, and as it's known, there is a self-specific to this antigen T lymphocytes, which might recognize this self-antigen, and this is a normal way of recognition of all antigens, even the self one. Then reactions which appears is different, and these reactions called tolerance, self-tolerance, and these reactions did not lead to activation of the cells, proliferation and differentiation, how it is in the first case, but to energy, tolerance, or deletions of the specific clones. And these are the main characteristic of so-called autoimmune tolerance or self-tolerance. And this is a characteristic for a different situation in the organism. Tolerance in the immune tolerance in the organism is appearing in autoimmunity, what we are talking about today, but also in pregnancy, where mother immune system tolerate the semi foreign or semi-self antigens of the of the baby. Then there we have gut intolerance, gut mucosal tolerance, which is some different characteristics, but it is very important, and it appears in the gut, and it is tolerance against certain most food allergens, and they did not induce the, inflammatory reactions, which is in the other case. Also, tolerance is so-called transplantation tolerance, which is induced by the doctors, and in when organ transplantation is appearing. So these are different types of tolerance, which are very important, as you see, for the function of the immune system and organism. This tolerance is consisted of two main parts. One is realized in so-called central part, or in the central immune organs, and this is thymus and bone marrow. And what is happening here? Here the cells are produced and educate to be T lymphocytes or to become immune competent. And in this, several processes are happening here, and these are apoptosis. We'll see a bit more later. Apoptosis, changes in receptors which recognize the self-antigens, different self-antigens, and development of regulatory cells, which is a set of cells which are very important. And then the cells are going in the periphery of the immune system, which means lymph nodes, spleen, and tissues, where another processes are appearing, and they are connected with energy, apoptosis again, and suppression, different level of suppression. So the tolerance, autoreactive tolerance, has two parts: central part and peripheral part of realization of this tolerance. In the thymus and in the bone marrow, as I told you, these central organs, what is happening is that in thymus, T lymphocytes are differentiated from the stem cells, and they are going different steps of this differentiation, which is connected with appearance of CD3, CD4, CD8, and other receptors characteristic for differentiation. But we are talking that in the same time, in thymus, under the influence and of so-called aerogenes, all internal antigens of the organisms are expressed on the stromal cells or on the other cells. And when T lymphocytes are recognized these self-antigens, they either go of negative selection, so-called negative selections, they are dying or fell in apoptosis. And if they recognize these self-antigens with a very high strength or with the very low strength, these cells are dying or going to apoptosis. And this only about 5% of all T lymphocytes which are differentiated in thymus going out or went out from the thymus, and they are in between. There are also self-specific recognized T lymphocytes, but after that they are going in the peripheral suppression mechanism, which are assured not a strong reaction against self-antigens. So the differentiation of T lymphocytes to recognize self from non-self antigens is happening in vast majority in T lymphocytes. Also, in thymus. Also, the nearly similar but not the same processes are going in the bone marrow toward B lymphocyte selection, and there are mechanisms which make T lymphocytes and T cell receptors, which are immunoglobulins, to recognize self-antigens expressed on the stromal cells, and cross-linking of these receptors lead to apoptosis. When this cross-linking is missing, these cells, immature B cells, goes in the periphery. No cross recognition here is they might recognize some B specific antigens of self antigens. So the first part, as I told you, the central part of peripheral of tolerance followed in the peripheral tolerance, which is which is realized in lymph nodes, the spleen, and tissue of the organism. And there are several mechanisms by which this peripheral tolerance is driven, and this is ignorance of the antigens, of autoantigens, which means do not recognize some antigens, split tolerance, energy, and suppression. All these processes are important because they're happening of different part, time, and concentration of the antigen. So these are factors which drive these processes. And we will see that what means ignorance of antigen. This is not recognizing self-antigens, and there are two reasons for this. One is that the antigens is autoantigens is expressed in so low concentration on the surface of the cells that this is under the sensitivity of the receptors which might recognize these cells. And the other phenomenon which is important is so-called sequestrated antigens, which are not expressed on the stromal or dendritic cells in the primary organs, in thymus or in B cell or in bone marrow. And these antigens usually are connected with some organs which are usually have a membranes, and they are not presented in the thymus. And this is some brain antigens, eye antigens on the lens of the eye, testicles, uterus, and the other one is the experimental model. These antigens are not presented. They are not going under the IRA genes, which helps to present these antigens in these primary organs. That's why they are unknown or ignorant for the immune system. And the immune system recognized, anti-lymphocytes recognize this tissue as a foreign antigens. That's why they do not express tolerance against them. The other phenomenon is so-called split tolerance. The antigens are recognized either by T cells mostly, or some part of them induced by recognized by B cells, and different concentration of antigens are needed to be recognized by these different cells. So in some antigen might be recognized by T but not by B cells, because B cells need more antigen expressed to be recognized. So this is called split recognition. To create or to make a full immune reaction, the first part is antigen presentation on the antigen presenting cells in the context of MHC antigen or HLA for human A or B, DR antigens. So they present antigens or epitopes on the surface of antigen presenting cells, and it is recognized by T cell receptor on the T cells. But it's not enough to induce activation of the cells. It's absolutely necessary to have a second signal, which is exercised by different adhesion molecules, and they are most often CD28 or B7, and the other one is for B cell signals, CD40 or CD40L. These second signals are very important, and if they miss in this case, you see, in this case appears apoptosis or death of the cells. There is no transfer of the signal toward the nucleus of the T cell, inducing activation. So this is one of the mechanisms of energy, which is characteristic. Missing of this adhesion signaling is characteristic for this energy. This mechanism is very important in this peripheral suppression, which is induced by so also depends on different conditions, such as when it appears, and it is characteristic for so-called neonatal tolerance. The other one is the way the antigen, if the antigen is presenting to the in the adult organism, but is not typical way. Say instead of through the mucosa or through the skin, put if antigen appears as an aerosolic, so inhaled antigen, or introduced by bloodstream, which is a different way, and it induces a tolerance. We'll see why. And sorry, the third group of important mechanism are T cell suppressor cells and a whole bunch of suppressive cells. We will see them. And another also it was very popular several years ago, this idiotype network, which induce between antibodies, and I will explain what I mean, between antibody on cell levels also, which is very effective, or which it is considered as a very effective suppression which is induced. But better is to say that it's a regulatory system. Neonatal tolerance, as I told you, when very early in the life of neonate is induced, put the antigen, then several weeks afterward, and this is a model which is very famous from Medawar, one of the fathers of the transplantation, and the clinic and the immunology at all last century in the medium. And this introduction of the antigens, say intravenously or through the skin in this very early period, leads to tolerance later, when another transplantation or the skin transplantation is made to the same animal, and the animal tolerate this skin transplant, but do not tolerate another skin transplanted for a different mouse model, which are not beforehand introduced to the same antigen. So it's it's known that in these first weeks of the neonatal life, it's the mouse are quite tolerant if the antigen is introduced there. Another way, as I told you, in adults, is when antigen is applied through the aerosol, and this and the same experimental model is with the induction of transplantation tolerance. When this antigen is introduced by inhalation, the mouse became tolerant and there is no reaction. The same thing is happening if the antigen, if the receptor, or some of the receptor of T lymphocytes, which recognize the antigen presented on the antigen presenting cells, such as CD4, CD8, B7, CD40, this adhesion molecule I just mentioned is as a second signal for induction of activation. So if this blocking of this receptor is done in the same time as the antigen is introduced, the reaction is not happening. This is a control with the PBS. So it's important to know that if you block these molecules which are helpful for the induction of activation, as well as we change the way of introducing the antigen, the tolerance is induced. And as I told you, the most strong phenomenon which are important for keeping this peripheral tolerance are suppression. And suppression might be exerted through cells or through network with the antibody and cells. And I'm sure you know the practicals. We have quite information about so-called T regulatory cells with the phenotype from CD4 positive, CD25 positive, and FOXP3 positive. These are cells which were identified after 2010 year, and it happens that they are very important in this suppression. They were found everywhere, in thymus, in peripheral blood, in lymph nodes, as you see here, in spleen, and the tonsil, and they are originating from the thymus, or what is now accepted, they originate from thymus and differentiate as a T cells there. But there were different hypotheses that part of them might differentiate in other places, such as gut mucosa. What is important, this phenotype of these T regulatory cells are, first of all, these are antigen-specific recognition. They are activated of a certain part of the antigens, and then they exert or they express their suppression, very strong suppression in all immune responses, such as T cell activation, T cell and proliferation, so on, CD8 positive cytotoxicity, autoantibody production. And these cells has a lot of receptors which were identified, through which they exert their immunosuppressive function. Why it's so important? Because nowadays these cells are a target, this receptor are target of different antibodies in aid to suppress particular autoimmune or cancer reactions. So you probably heard about this PD-1 suppressor receptors, which is a very popular now as a target for the target therapy is some cancer. We will have a lecture and speak more about this. Very strong suppressor effects might be exerted through the CTLA4 receptors. These also are important receptors, and against all of them already have there are monoclonal antibody which targeted these receptor cells in aim to increase their action or decrease their action, and induce some more suppression of autoimmunity, or they repress function of T lymphocytes, or these T regulatory cells in cancer. In cancer, they must be activated or induced more function there. But while in autoimmunity, their function is the proper function is to suppress this population. As you see, they might produce a lot of interleukin also, and they're HLA specific. There is a cell-presenting TCR receptor which recognized autoimmune. Most often they are alpha beta, this receptor. So these regulatory cells are the how to say the king of the new century in immune regulation, and they're already used not only to explain a lot of reactions in this situation such as autoimmunity or cancer, but also they are used for production of a lot of these new biological agents which are used for treatment. But they are not the only one. There is a lot nowadays identify relationship between T regulatory cells and other cells, or characterized another sets of cells which are taking part in the cell regulation of the immune response. I'm not going in deep, but you see these are in the field of antifungal and autoimmunity. This TH17 subsets is very important in the intracellular pathological of autoimmune disease, in all areas of the defense or the immunity against different situations. So T regulatory cells and a bunch of not very properly, how to say, nowadays characterized other mechanisms which are connected with the suppression in peripheral tolerance. And the other type of suppression, as I told you, is so-called, just a moment, please. So-called idiotype, anti-idiotype regulation. Why I am telling it? Because you have heard in the internal medicine therapy that there was very popular using intravenous immunoglobulin therapy introduced to different diseases and autoimmune diseases in between. So what is idiotype? Idiotypes are epitopes on the, if this is immunoglobulin FIB specific recognizing part, there is not only recognition for the foreign antigen, but also there are epitopes which on this internal or external part of this immunoglobulin part, which are called idiotypes, and there are a lot of them. And these are proteins, epitopes. They are inducing production of antibodies, specific antibody against them. These are also has epitopes, and there is antibodies which are against this parts of these immunoglobulins. So if they recognize each other and make a net between, at the end result is that this idiotype, anti-idiotype network make suppression or make regulation is most is better to say regulation of disturbed autoimmune clones or a phenomenon, and this is the base for the usage of this anti-idiotype of this immunoglobulin for intravenous application as a regulator of some autoimmune diseases. They might activate, not the most pronounced action is tolerization or tolerance suppression of the pathological clones which are appearing in some, especially autoantibodies, which appear in different diseases. So if we make a, how to say, overview, the autoimmune response is there is autoantibodies, autoantigens in the healthy organism. There is T lymphocytes, as I told you, 5% are going in the periphery, but all these two participants of the immune response are subjected to different suppressive mechanisms which prevent them to go to autoimmunity as a process, as a phenomenon, or autoimmune diseases as a clinical expression of this condition of autoreactivity. But if some of these these mechanisms of tolerance are bypassed, then the autoimmune response appear. And this different dysregulation might appear on several levels, and we just talk about them. This is dysregulation of idiotype, anti-idiotype network, increased or aberrant cytokine production in inflammation. Usually we'll talk about a bit later, or impaired apoptosis, which also is important mechanism which might bypass these regulatory mechanisms and the autoimmunity to be to escape autoreactivity, to escape and become the autoimmunity. So this peripheral tolerance and these regulatory mechanisms are very, very important. So if this is the theory, or how to say, the basic of induction of autoreactivity is a normal, as I told you, process, but induction of autoreactivity is also pathology and leads to autoimmune diseases. That's where it is. Different autoimmune diseases may appear, and this is connected with the autoimmune pathological regions. And this is autoimmune antibodies or specific autoimmune cells. Both parts of the immunity might induce pathological processes which destroy normal tissue. Autoimmune diseases can be organ specific. It's appear usually in one against one antigen of particular organ, and might be systemic if these antigens are widely distributed in the organisms, such as connective tissue diseases, or these are systemic autoimmune diseases. And these diseases are some about who induced these diseases. A lot of work was done, let's say nearly century, how these autoimmune diseases appeared. And it is now known that, well known, that there is a multifactorial reason for induction of this autoimmunity, and they are mainly connected with genetic disturbances on HLA or another parts of the idiotype, or also epigenetic factors which are connected with the methylation, with the biochemical processes which might induce a different antigen. Another very big field of factors which are the reason for induction of autoimmune diseases are environmental factors, such as the increased pollution or industrial pollution also, which infections, which are very important group of inductors of the autoimmune diseases, because the organism or the immune system might cross-reactive recognition may recognize wrong between bacteria or virus antigens and self-antigens. Another very important area is a hormonal system or endocrine system, which all are connected with the strength or induction of different reactions, and increased estrogen or decreased testosterone or vitamin D are very important factors which are identified already. Immune deficiency status as an underlying condition is very important also, because it was determined that most of the autoimmune diseases have some other immune deficiencies, such as decreased IgA, decreased T regulatory cells, complement defects, and so on. We'll have a little bit more later here. So here I found some slides which summarize the importance of these factors, as I just told it for you. So the genetics predisposition is well noticed observation from usually at the beginning in the families, that there is a family which has more often incidents of certain autoimmune diseases. But nowadays, these factors or genetic factors are more or less identified, and they are connected not only with HLA, but I am showing you with HLA alleles, which are connected with different diseases. The risk of induction, at higher induction in of certain disease connected connected with HLA defects, is expressed by so-called relative risk, and it is seen here. So bacterial disease or ankylosing spondylitis is very how to say, it's very often appear on the in patients who has HLA-B27 positivity, and you see how high is this risk. More often it appears in men, one of the small number of the autoimmune diseases where men are more affected, but see how much the risk is in when you have B27. Also, acute anterior uveitis is connected with B27. The Goodpasture syndrome, this autoimmune glomerulonephritis, is with DR2 connected, and quite, you see, nearly 16 times more often when the patients or the person has DR2, is more often appearance of this disease. Multiple sclerosis is connected with DR2 also, but no so pronounced risk about this. And Graves' disease, thyrotoxicosis, is with DR3. But also myasthenia gravis is another systemic autoimmune disease. This is not systemic. These are connected with DR3. And systemic lupus erythematosus is connected with DR3 also, about 6 times risk, relative risk. And you see how much more often it affects women compared to men. Rheumatoid arthritis, DR4 is connected, and now it's known that not the whole DR4, but some suballelic gene defects are important, and so on. What is important, as I told you, that this is it looks very horrible, but it is the reality that not only HLA is connected with certain HLA alleles, but also other antigens, and these are diseases, and these are identified genes and which are connected with different diseases. But what we'll see, one and the same genes might be connected with autoimmune, but also in other diseases. And if you have, we don't have now time to look proper, but the important conclusion is that not only HLA system is has defects, and it's important for autoimmune diseases, but also combination with the gene defects not always identified their importance, but they are also found that they might be connected with such disease. Autoimmune and not autoimmune diseases. Immune deficiency are associated with the other autoimmune diseases. Just a moment, please. Somebody is very tedious here. Не мога да говоря сега. Не мога да говоря, в лекция съм. После ще те звънна, ако може. Извинявай. So there are some of defects which are connected with different other genes which are connected with the autoimmune diseases. And you see C1q defect, knockout. So complement deficiencies are also important, CD4, CD3, and other ARE, these were the genes which are important for the presentation of autoantigens in thymus. So defects in this area is very important as a base for the development of different autoimmune diseases. And this is also, we should know that some defects are identified as gene defects, other are identified as diseases already, and mechanisms which are acting as a factor predisposed or make difficult, more strong, how to say, expression of the disease. So, as we already mentioned, that the sex and the hormones are important, and there are a lot of autoimmune diseases which are more characteristic for the women. And on the first place, these are systemic lupus erythematosus and other systemic autoimmune diseases on the connective tissue. And see, for lupus, it's very pronounced. Nearly 90% of the patients are women. Sjogren, also very high prevalence of women. There is a different expression. Most often prevalence is in women, but there's also some diseases which has nearly the same, men and women. One other reason is the influence of surrounding or environment, such as infections, some drugs, some medicine, and other products which might induce creation of so-called neoantigens. So if this is the antigens, normal antigens, against which there will not be autoimmune reaction, and this is the normal we already talk about, but combination or changes in these normal antigens with different bacterial group or bacterial antigens, drug chemical antigens, and so on might induce so-called neoantigens, and these normal otherwise antigens are recognized as foreign antigens, and they induce the immune reactions. And in this area, very high homology were identified between some bacterial antigens, such as Shigella with B27, or viral antigen Klebsiella also has a very high with B27 with this rheumatoid arthritis, and viral antigens such as Coxsackie virus, which is it has quite a high homology with myocardial antigens. So there was one hypothesis that Coxsackie B is connected with induction of autoimmunity of the myocard. And so you see, Epstein-Barr virus or Escherichia coli also has a very high homology with some antigens which are characteristic for certain diseases. And nowadays, this homology with Epstein-Barr, some antigens of Epstein-Barr is connected with the pathogenesis of rheumatoid arthritis, where these antigens are thought that there is a mixed cross in the beginning, mixed cross presentation of this bacterial or viral antigens in induction of autoimmune condition. Here are the importance. This in the recent 20 years, it was very high proven that different infections has a role in induction of autoimmune diseases. It was noticed a long time that some infections might be in the beginning or several times before induction of autoimmune, the beginning of autoimmune disease. So nowadays it was proven that there are several processes which are connected with this. One of it is molecular mimicry. I'm sure you know that this mimicry lies in the beginning, in the basis of this cross recognition, wrong cross recognition, and induction of the autoimmune processes. It is one of the popular hypotheses for induction of on the influence of infection on the induction of autoimmunity. Microbial superantigens such as lipopolysaccharides, which is very often in the surface of bacterial or viral bacterial antigens, is recognized as a polyclonal T cell activator through the so-called super antigen recognition, which is a different mechanism from the usual one. The recognition of antigen is not through the the HLA presentation and recognition of T cell receptor, but by other outside recognition, which is called superantigen recognition, and this make a polyclonal activation of a lot of T lymphocytes. It's not necessary to have a specific receptor about this recognition. That's why might activate also autoreactive T cells. Also, often infections, even chronic infections or acute infections, are connected with production of cytokines, and they change the environment around the immune cells and might induce antigen presenting cells such as gamma interferon, might induce presentation, increased presentation by antigen presenting cells through induced expression of HLA and make them non-professional antigen presenting cells, and two other processes which are molecular processes identified are connected with the bacterial, with the infections. They call epitope spreading or bystander activation. These are processes which are connected with the increased presentation or recognition of antigens by T cells, and false induction of reaction might happen to false induction of autoimmune reactions instead of reactions to the environmental antigen. So different mechanisms are responsible for the tissue damage. So until now, we were talking about the immune processes which are important to induce or realized autoreactivity, the pathological reaction against self-antigens. Now, nevertheless, what mechanism, or if we make a overview of the mechanisms which might induce this damage, humoral factors. These are antibody or immune complexes between antigen and antibodies, which might induce so-called type 2 or 3 hypersensitivity reactions, which leads to cytotoxicity and damage on the tissues. Cellular reactions such as delayed type hypersensitivity or cell cytotoxicity reactions are characteristic as a mechanism in autoimmune diseases of the nervous system, such as encephalomyelitis, syndrome of Guillain-Barré. But these are cellular mechanisms connected with activation of T cells and secretion of cytokines, which are very disturbing for the normal tissue. Mixed mechanisms means that antibody or immune complexes with combined with cellular factors, and they are inducing pathogenesis of some dermatomyositis, thyroiditis, Hashimoto, and diabetes. So they have specific autoimmune antibodies and T-specific T cells. And this regulatory mechanism, this regulation of this most often activation and suppression or autoreactive suppression are connected with other diseases. And these mechanisms, we can see they are mediated by T cells, by B cells, and antibodies, and these are the systemic lupus erythematosus. You see in the pathogenesis take part T cells, B cells, and also characteristic autoantibody, antinuclear antibody. In diabetes, pathognomic, so most important damaging mechanisms are T cells, but also B cells are taking part. Myasthenia gravis also helper cells, B cells, and pathological antibody, which are known as a pathognomic antibody, anti-cholinesterase receptors. We'll see a little bit more. And multiple sclerosis also, T cell, mainly T cell pathogenesis, but also disturbed presentation and not known characteristic antibody. Nevertheless, which is the inducing immune mechanisms? One of these just we are talking. The inflammation, the development of disease going toward chronic inflammation, and this is activation of platelet system, other complement system, and other macrophage activation. And all these phenomena are connected with production of different cytokines and lead to a chronic inflammation, then sclerosing, and that of the specific tissue and the functions which it present. And once induced, the autoimmune diseases are going as a perpetuum mobile, not going back. Might be their follow-up might by the treatment might be modulated, but the final eradication of the disease at the moment is not real results. Epidemiology of autoimmune diseases is how to say important, because these diseases are increasing all the time. More than 100 identified autoimmune diseases and conditions which are not all defined as an immune disease, but has quite features of autoimmunity. Mean world incidence is about 5% from the population, but in certain countries, these are countries with more highly industrialized countries, which means the influence of environmental pollution, the incidence is higher, and it reached 10%. Then there is a wide world tendency of increase of autoimmune diseases. So we should expect next year with better diagnosis, but in the condition in which we live, to have increase of the autoimmune diseases. They are characterized or classified as systemic, which are connected with the widespread autoantigens, such as autoantigens from the connective tissue disease, and organ-specific diseases, which are connected with particular antigens in a certain organ. So this is one of the most popular classification, and in the medium of the classification, there are different certain disease which have reflection on the whole organism, not only on the certain one organ. This is the more how to say, nowadays classification, which reflects not only systemic or organ specificity of autoimmune diseases, but also clustering of some diseases, and that's why this classification is called cluster classifications, where in certain, and it might be related to to genetic background, where some diseases are appeared more often in certain family, and this might be such as a combination of thyroid autoimmunity, which is Graves and Hashimoto, but also autoimmunity toward other endocrine glands, such as against Addison disease and so on. And here is a clustering of systemic autoimmune diseases such as lupus, primary Sjogren syndrome, and polymyositis. These are very often that's quite often appearing in one close relation families. So this classification is connected with genetics and with the incidence of these diseases. So here we have diseases which might be transferred to the neonates through the during the pregnancy, and these are diseases which are most often connected with the autoantibodies which of IgG class, which goes through the placental membrane, and they may appear and even express a disease in the fetus, in the neonates. And diagnosis of autoimmune diseases are based on clinical symptoms, increased serum IgG, specific autoantibodies, decreased in serum complement, which C3 and C4, which means activation of the complement system through the classical pathway, decrease of T regulatory cells, which I already explained to you what is the importance of these T cells, and the diagnosis is must be confirmed by biopsy of the target organ, which is the last and more sure diagnosis. Now, a little bit examples about the role of nuclear antibodies in the disease of systemic autoimmune connective tissue diseases. Anti-nuclear antibody detection, you have the practicals on this. You know how they are already tested. There is several way of testing it, and they are characteristic for these diseases. Most importance they have in systemic lupus erythematosus, but also in progressive systemic sclerosis or scleroderma, a mixed connective tissue disease, where nearly 100% of these diseases are positive for anti-nuclear antibody, Sjogren syndrome, and where a certain type of autoantibody, anti-nuclear, are characteristic, and we will in the next slide. Half of the rheumatoid arthritis have anti-nuclear antibody, but this is not the most diagnostic one for rheumatoid antibody, and so on. Discoid lupus, this also has a high incidence of antibody. But anti-nuclear antibodies are a bulk of antibodies which are directed against different antigens of the connective tissue disease, and they are characteristic for different diseases, the specificity. So as you see, nearly all diseases are connected with, so nearly all specificity might be met in systemic lupus erythematosus. So you see anti-LA, RO, SM, and so on. But some are characteristic only for one. Double-stranded DNA are pathognomic for systemic lupus erythematosus. But anti-SSI or LA and SSB are characteristic also for sclerodermia and syndrome of Schönlein, which are important. Antihistone antibodies are characteristic for drug-induced systemic lupus erythematosus. Anti-U1 RNA antibodies are mixed connective tissue disease, and nearly 100% of the patients has this anti-U1 RNA specificity. Centromere antibodies are characteristic for CREST syndrome, which is localized sclerodermia, and so on. Anti-Rho and anti-Jo antibodies are characteristic for Sjogren or polymyositis. So these specificities are helping to precise to the diagnosis of systemic diseases. And one example here for systemic lupus erythematosus, as we talk, they occurred on a certain part of the life between 20 and 40 years, but it might appear also in children or in much older people. 90% of the patients are female. Autoimmune reactions is against double-stranded or natural DNA antigens, and these are pathognomic autoantibody, which prove or consider as a pathogenic factor. And mortality after the beginning of the disease is about 5% per year, increased, of course, and the characteristic symptoms are facial rash, and these are butterfly patient, but not only this rash is characteristic, also oral or nasopharyngeal ulcers, fever, arthritis. These are the most pronounced symptoms, but also different serositis are characteristic, which is pleuritis or pericardial fluid. Very often about half of the patients appear as a first presentation with renal disease, which leads to renal failure. Central nervous and brain system and brain are also involved with seizure or the psychiatric diseases, symptoms, and also peripheral vasculitis are characteristic for these patients. What is important, and as a management, these patients should be treated with, it depends very much how heavy and how long this disease is going, starting with non-steroid drugs such as aspirin or ibuprofen for the light condition, but then goes to corticosteroids and even bolus, even the heavy courses with the corticosteroids. But nowadays, and systemic lupus also, but and other systemic autoimmune diseases are subjected to this so-called biological targeted therapy, and they have a very, very faster and a bigger effect. And it's important to avoid some factors which might exaggerate the disease. Characteristic for and very helpful for the autoimmunity of the liver is testing the antibodies, and the diseases which are connected here is autoimmune hepatitis type 1, type 2, classical, and the other one, and overlapping syndrome, which is important, and overlapping is between primary biliary cirrhosis, which is another autoimmune disease of the liver. And these are antibodies which are which have importance. These are anti-nuclear, anti-smooth muscle antibodies, and anti-actin antibodies, and so on. So antibodies are in liver autoimmunity are very important rate to prove the diagnosis, and to make a more proper approach to more effective treatment. And here it is an anti-nuclear antibody. I'm sure maybe you have seen them on practical anti-nuclear, which is these are indirect immunofluorescence, and these are for anti-nuclear antibodies. These are hepatocytes, and their nucleus are positive fluorescentin. Anti-mitochondrial antibodies are important for primary biliary cirrhosis, and these are renal tubule, which are positive for this diagnosis. LKM1 antibodies are also characteristic for type 2 antigen, and these are anti-smooth muscle antibody, which are characteristic for hepatitis 2 type 2 autoimmune hepatitis. These are examples how this antibody of IgG class are going through the placenta, and they can induce thyrotoxicosis in a baby. So this lady has Graves' disease, and this antibody against TSH receptor are inducing this hyper function of thyroid glands. Another characteristic and identify pathogenic antibodies are anticholinesterase antibodies, autoantibodies, which are IgG auto antibodies against antigens of cholinesterases, and these antibodies block the receptors for cholinesterases, and do not permit these cholinesterases to go in the muscle, so they block the signals from nerve to muscles, which is the problem in this disease. Anti-glomerular basal membrane antibodies are connected to characteristic for Goodpasture disease, which is autoimmune glomerulonephritis. The antigen, the pathological antigen, is distributed through basement membrane in the glomerulus, and here is a positive reaction of this antibody in direct immunofluorescence of the liver, of the renal biopsy, and these are the experiments which proves that these antibodies are pathognomic for the development of this autoimmune glomerulonephritis. Another type of immune disturbances is systemic lupus erythematosus, is induction of sclerosis and so-called lupus nephropathy, and losing the function of the kidney because of how to say, creating in situ immune complexes between anti-DNA, double-stranded DNA antibody, which are they're stopping in the glomerulus and induce chronic inflammation, followed by sclerosis of these glomerulus and renal failure. So this is characteristic for the chronic glomerulonephritis, but also for progressive, diffuse progressive proliferative glomerulonephritis of lupus. Lupus nephropathy might have a different type of mechanisms which induced liver failure, renal failure. The pathogenesis of rheumatoid arthritis, which is another very wide, most often appearing disease of systemic autoimmunity, and the mechanisms of induction of this disease is not as for the most of the is not very clearly described, but it's known now that the main pathogenic mechanisms in rheumatoid arthritis is T-cell dysregulation connected with cytokine, pro-inflammatory cytokine dysregulation, increase of this cytokine production, and which leads to induction of activation, quite a complex activation of B cells also, not just dysregulation. B cells which produce autoantibodies, which are pathogenic, and they are in the basis of tissue damages here over production of this autoantibodies such as rheumatoid factor, different isotypes, or anti-CCP antibodies, which are autoantibodies against different citrullinated proteins. So the pathogenesis here is combined. There is quite a significant part of T cells activation, the T cells dysregulation, T cell activation and production of pro-inflammatory cytokines. But on the other hand, the mechanisms which are connected with fibrosing and sclerosing of the ankles, of the tissues. And in the same time, there is a quite useful for diagnosis autoantibodies such as rheumatoid factors, different types, anti-CCP or MCV antibodies. So the very complex pathogenesis of rheumatoid arthritis, but there is quite a good markers to identify this antibody, this systemic disease. And the principles in the therapy of autoimmune diseases as I told you, there is a increasing strength of the used therapies. If we start in the beginning with control of metabolic insufficiency in organ-specific diseases, these are introduction of different medical substances which improve the function, the lost function of these organs, such as thyroid thyroxine or other medicine. The other rate or the other level of therapy is the first level, non-steroid anti-inflammatory drugs, because, as I told you, this immune-induced inflammation needs to be suppressed, and start with the non-steroid, then going to steroids, different steroids, immunosuppressive drugs. Methotrexate in rheumatoid arthritis is very important. Then the plasmapheresis was one of the approaches which in this case means to evaluate pathological autoantibodies and high protein overload of this autoantibodies, so they repress the immune regulation and induce a better condition in these patients. In some diseases, splenectomy might be useful, and nowadays it's a very big field for introduction of this biological agents, which are mostly a monoclonal antibody and target therapy against different elements of the increased immune response. The target is immune suppression usually. And here there are some of these biologicals, which are, as I told you, a monoclonal antibody against receptors, against co-stimulatory molecules, against cytokine receptor, in term to induce more co-stimulatory process to increase or to improve antigen activation of the T cells. This induction of activation of T regulatory cells and antigen clearance through the immune complexes. So these are immunotherapy or field of immunotherapy, which is increasing the meaning of last years. I think this is the last one. Please, do you have questions? I don't know, maybe you're here already or not. Eho, please. Do you have any questions? You don't have questions, maybe you're not here anymore.
Doctor, we're writing in
Yes.
the main chat and we're saying that everything is clear. So thank you very much, really.
Okay. Have a nice day. Next
You
week,
too.
I hope we will have a successful lecture also. I am going to present you something about the immunology of the cancer, of the tumors. I think it will be interesting for us, for you. Okay.
Perfect. Yeah, it sounds very interesting.
Yes, I hope we will. I hope it will be not so heavy for you as was today, but autoimmunity is quite a big a point. I was planning to have another lecture also about the disease, algorithms of these autoimmune diseases, but it seems we'll not be able to do this. So I will present the tumor immunology, okay, next week.
Okay, perfect. So
Have
see
a
you
nice
next
day.
Monday.
Yes, see you next Monday. Sad vakar bija štāvot.